Okay, I think maybe we have a full complement now. So good afternoon. Welcome to our symposium with Dr. Joanna Shisler, who has kindly agreed to speak to us about funding opportunities at the National Science Foundation. I just want to give a brief introduction to Dr. Shisler for those who don't know her, and then we can let her take it away. So Joanna is a professor of microbiology and associate director of water and health research at the Safe Global Water Institute at the School of Molecular and Cellular Biology at the University of Illinois. She earned her BA in microbiology in 1990, followed by her PhD in immunology and molecular pathogenesis from Emory in 1996. And she did then postdoctoral work at the Laboratory for Viral Diseases at the NIH until 2001 before joining the faculty at the University of Illinois. And her own work focuses on viral pathogenesis, focusing on environmental persistence, immune system evasion, and mechanisms of viral spread throughout the environment. So, and interestingly, Joanna has taken a leave of absence from her work to become a program officer at the National Science Foundation, extending through February of next year. And she has kindly agreed to speak to us about the NSF in general, and specifically about funding opportunities in immunology and microbiology. So, Joanna, I want to personally thank you again for working with us on this date and your presentation. Welcome.
Thank you. So I need to give a few caveats before I start. The one is that I cannot see any of you because of my screens, the way I'm set up. So if you have questions or want to interrupt me, you'll need to just speak up. I'll stop the slideshow. I only have about 15 slides, and then I should be able to see you.
So Joanna, what I can do is actually, if asking everybody to just type in your questions on the chat, and I'll be happy to sift through those for you. Okay.
Great. So what I want to start or preface this with is that this is the initiation of a conversation. So I liken this to each of you traveling to a foreign country where they don't speak English, and that you need to start learning the language and start understanding the culture. And this is that first pass. And so to start things off, this first slide shows a picture of the National Science Foundation building. And I want you to understand what our mission is, because it's a little bit different than the mission from NIH or USDA or the Gates Foundation. And that's important for you to think about, because when you think about how to frame your proposal, you really want to think about what the mission is here. And so the mission here is to support fundamental research across virtually all fields of science, engineering, and education. Now notice what it doesn't say is to support biomedical research that can help human health. We're interested in supporting fundamental research. And so that's something to think about when you apply to NSF, and we can talk about that later. I included a couple of little facts about NSF. So I want you to understand what our budget is. It's $8 billion. So that is much smaller than NIH's budget. But we still get quite a lot done. Most of our budget is committed to basic research and education. And I'm going to talk about why I mentioned research and education later on. You can see that we put a very strong emphasis on training, especially graduate students and undergrads. That's part of our broader impacts, and I'll talk about that later. We award about 12,000 proposals a year. We've had a rich history and excellence in research. We've had about 240 Nobel Prize winners who've received funding, including Jennifer Doudna, who just received the Nobel Prize last week. So within the NSF, there are different directorates, and the ones that you would be probably most interested in are the Directorate for Biological Sciences and maybe the Directorate for Engineering and Computer Information. But I want to talk about just the Biological Sciences Directorate, okay? And so what's our mission? Well, it's to enable discoveries for understanding life, advance the frontiers of biological knowledge, increase our understanding of complex systems, and provide a theoretical basis for original research. So once again, I want you to see that what we're interested in is understanding discoveries to help us understand life and that is including humans and mice and C. elegans and Drosophila, but it is also much richer. It includes things like corals and other non-model organisms, wasps, squids. And so really it's about exploring basic questions in research. And I'll get to that in a couple more seconds. This is a slide that I feel like is really important because it shows the structure of the Directorate of Biological Sciences. So I want you to think about the directorate as a college, and within that college there are 4 divisions, and I think of those as departments. There's Division of Biological Infrastructure, and that's important for research resources, and I don't think you guys are being most interested in that today. What I think you'd be most interested in are these 3 other divisions. The Division of Environmental Biology, we call this DEB, the Division of Integrative Organismal Systems, we call this IOS, and the Division of Molecular and Cellular Biosciences, or MCB. So what I want you to notice is that these are at the top of the flow chart, and I want you to pay attention to these first. Don't read all this other stuff underneath. These are the main separations or organizations of this directorate. And I'm telling you this because it's going to help you understand the language of NSF, which is the first part in understanding the culture. So what does this mean? Environmental biology, integrative organismal systems, molecular and cellular biology. Well, basically when I think about these 3 divisions, what I think about is scale. So if you look at first this division of molecular and cellular biosciences in the green on the right-hand side, that makes a lot of sense to us, right? We're thinking about things that are happening on the submolecular or cellular basis. On the other end, in the blue, you have the division of environmental biology, and that's talking more about ecosystem, ecology, and evolution. And then what you see here is the division that I'm in, and it's in the yellow, and it's called Integrative Organismal Systems. And I think of us like the cream filling in the Oreo cookie. You can't see us if you look at the cookie from the top or the bottom, but we're in the middle, we're hidden, but we're the best part of the cookie. And we're really interested in studying the organism as a whole. And what I want you to understand is that there is some overlap. So once again, here's molecular and cellular biosciences, and they're interested in things like submolecular processes, regulation of genes, cytoplasmic organelles, but they can do research that goes up to the organism. With integrative organismal systems, we're interested in identifying how an organism is interacting with its biotic or abiotic environment, but we can work with things on the cellular level and up to the community level. And then environmental biology is looking at things like the population, the community, the ecosystem, and the biosphere. So we're going back to the structure here, and you'll see on the left-hand side, I said, what about immunology? How are you supposed to know where to go if there's nothing that explicitly says immunology here? And so what I want you to do is go back to this idea where I said we're interested in fundamental biology. We're interested in hearing what your question is. And if your question is, how do cytotoxic T cells release their granules, then maybe this is something that would be important to the division of molecular and cellular biology. If you are interested in asking, how does the immune system interact with an infection during disease, or why are some viruses mutualistic versus parasitic, looking at it with respect to how these viruses interact with the organism, then you'd be putting things in integrative and organismal systems. If you're interested in understanding the ecology and evolution of the immune system, or the ecology and evolution of infectious diseases and how diseases spread, then you'd be more interested in the division of environmental biology. So I'm giving you this framework so that you understand that really what you want to ask is, what's the question that you're interested in? What's the framing of your scientific program? Because that can help you decide where you're going to start talking to the NSF for funding, okay? So there's one other thing I want to show you in this slide. You know, I talked about this higher level here, and then you see within each of these levels, you can see that there are smaller square boxes underneath each level, and these are called clusters. Okay, so you have your directorate, that's like a college. You have your divisions, that's like a department, and then you have these clusters here. Okay, and these different clusters then are the focus of the science. So let's just walk through molecular and cellular biosciences, because I think that's probably most straightforward. You have a cluster of cellular dynamics and function. So you can imagine they might be interested in things that are subcellular. So how do organelles work, for example? Genetic mechanisms, well, that means that they're interested in understanding things like how are genes regulated on a molecular basis, or how are transposons or plasmids transported. And there's also molecular biophysics and systems and synthetic biology. In iOS, there's some clusters that you probably won't be that interested in, because they include things like behavioral systems. And so that's not talking about human behavior, that's talking about animal behavior. So for example, what is the molecular basis for tree frogs kicking out their legs when they're ready to give a mating call? Developmental systems, that has to do with how do organs or organisms develop, and that can include using things like C. elegans, but it can also include things like root development of plants. Neural systems is looking at how do neurons work within the context of the organism. What I'm in is physiological and structural systems. I'll talk about that in a few minutes. And then we have plant genome research, which is about sequencing plants that are important in agriculture. You're probably not going to be relevant to you guys. And then if you look at division of environmental biology, there are clusters like ecosystem science, which is looking at continental processes, so that's probably not relevant. What's more relevant to you might be evolutionary processes, you know, understanding how the immune system evolves over time, and maybe population and community ecology. So that would be perhaps understanding differences in immune systems across populations. So because I'm from iOS, I'm going to spend a little more time talking about that, but I want you to just get the landscape here. So let's talk about this integrative organismal systems, right? So what this shows you is just some of the research themes spanning from small to large organisms in hot and cold environments. So as you'll see, of course, we're interested in microbiology and immunology. We're also interested in things like hummingbirds and how they feed. With respect to microbiology and immunology, we're interested in honeybees because they have a very simple 5-member microbiome of the gut, so we can understand how microbiomes may form in humans. This is showing some of the work that we do in our neural systems cluster. And then we also work in things like development of animals, how animals respond to stress, such as changing environments. And then we also have research that looks at plants and how they change with environments. And that's probably not as important to you guys. So within iOS, what I think is probably most relevant to you guys is this physiological and structural systems. And I wanted to show you a little bit more about that. So within this cluster, we have a couple of different programs. And the one that's probably most related to what you're interested in is symbiosis, infection, and immunity. So we're interested in how an organism and a microbe interact with each other. And I know that we always think about, or I as a virologist always think about an acute infection, but that's not always the case. There can be chronic infections, there can be latent infections, there can be persistent infections, and there can also be mutual infections. And so the question is, well, what are the determinants that are important for regulating those different outcomes of infection? We're also interested in the microbiome and how the host and the microbiome interact. We're also interested in immunity and immunology with respect to how that has evolved. And we're also interested in other things like origins of life, especially when it comes to the immune system, or even to things like how mitochondria evolved, right? So what I want you to see is that there's a lot of different things that we're interested in. In addition to symbiosis, infection, and immunity, there are other programs like physiological mechanisms and biomechanics, and that looks a lot at mechanisms of resilience of animals, and integrative ecological physiology, which is more about how the organism interacts with the environment. And so there's some eco-immunology that's in there that might be of interest to you. With all these, though, we're really interested in getting to the mechanism and in causation and understanding how your finding will be relevant to non-model systems. So what are you doing where somebody who studies the human immune system might all of a sudden be really interested in the paper that you've published and how this might apply to human disease? And a lot of the times, a lot of the ways people choose to do this is by using comparative biology, where they have two systems that have different phenotypes, and then they can identify the basis, the mechanistic basis for that difference in phenotypes. So that's iOS. I just want to mention briefly a couple of things about this division of environmental biology and molecular and cellular biosciences. For the division of environmental biology, I think what you should be most aware of is a project called Ecology and Evolution of Infectious Diseases. And this is something that you can find on the DEB website. So you would do a Google search that says NSF, DEB, and EEID, and you would find the program officers that you should contact who could give you some more information about this program. And then the other thing I want to talk to you guys about is molecular and cellular biosciences. And so this is a group that supports quantitative, predictive, and theory-driven research to understand systems at the molecular, subcellular, and cellular levels. And so once again, you could, for example, be interested in understanding how gene expression is regulated in T cells that are activated versus non-activated, and that might be something that genetic mechanisms was interested in. So there are lots of different ways that immunology and microbiology research can be supported at NSF. It all depends upon the question that you're asking. The worst thing in the world for you to do when you submit an NSF grant proposal is to have your grant start off, 3,000 people died this year from disease X, because that's not what we're interested in. We're interested in what's the fundamental biological questions you're wanting to ask. And so that relates a little to my next slide, which is NSF merit review criteria. We're very different from the NIH in that we have two different merit review. We're not only interested in the science, and that would be the intellectual merit, which is on the left-hand slide here. We're also interested in broader impacts, which is on the right-hand slide. So let's just talk about intellectual merit. You know, the questions that the panelists will look at is this research going to advance knowledge in and across fields? Are the investigators appropriate? Do they have the training? Is there an originality to the idea? Is this potentially transformative research? But broader impacts is something different. It's more than just saying, hey, I'll support a graduate student or two. It's really going to that next step to promote science. And that can be promoting teaching, training, and education. It could be enhancing infrastructure for research and education. But a lot of the times it's community resources and outreach. So there are multiple PIs who have created apps or made board games or collaborated with museums to get the lay people to understand and become more literate in science. It also includes things like participating in underrepresented groups and showing how this research can be beneficial to society. So the last 3 slides I have here are just some things I think you should know if you want to look into NSF further. The first is that each of our divisions have virtual office hours. I would suggest you attend these virtual office hours because there's listings and discussions of new and ongoing funding opportunities, topics of general interest. You can meet program officers and ask them questions. And so for that, for example, you'd want to go to Google and type in NSF and then iOS, and then go to the iOS webpage and look for virtual office hours. And you'll see an announcement. You can sign up for it by email so that you'll get a monthly, it only happens once a month, it's only for an hour, but you can get a monthly recurring nudge to tune in. And it's a good way of learning about NSF. And then I think a lot of questions that you guys will have is, what about medical research, right? Because that's what you're doing. Well, we're a little bit different from NIH in that we're not interested in research with disease-related goals. And what I mean by that is that we're not interested in diagnosis or treatment of human beings or animals. That's not normally supported, although in the time of COVID-19, we have an exception here because this counts for all hands on deck. And we can talk about that either today or one-on-one on how to think about submitting an immunology or microbiology proposal, including COVID, to NSF. We're really not interested in animal models to develop or test drugs. That's not what we're interested in bio. However, if you're an engineer and you come up with some method for being able to develop or to be able to inject drugs in a novel way, that would be something that engineering would be interested in. Research in bioengineering or information technology is something that we can talk about, but really that would probably be in computer information. And then there is some bioengineering and assistive information research, technology research to aid people in disabilities. That's also eligible. But really what I want you to take home is that we're interested in the biology behind a function that you're studying, not the disease itself. And we can talk about that a little bit more because it's a nuance and a lot of people get tripped up until they think about it for 3 or 4 times. This is my last slide. So what should you do next? If you think that NSF might be a good home for you, go find a colleague who has NSF funding and ask to read their grant, because you want to understand the culture of NSF. And the best way to do that is to read a proposal that's been successfully funded. Like NIH, we have an NSF award database that you can search to see what types of immunology and microbiology proposals have been funded. It will not give you the entire grant. It will only give you abstracts, but you can then reach out to those PIs and ask if they'd be willing to talk to you about their experiences with NSF. Also, you can start reading through this NSF Bio website to become familiar with funding opportunities. So what I just gave you was a crash course in learning a different language, and that should help you start going through the NSF website. And I have to apologize. It is a very dense and thick website. It's not very user-friendly, so you're going to have to be patient in learning. And then finally, reach out to program directors. We're pretty nice. We're a little busy, but we'll fit you in. And really, you should not be surprised if it takes you talking to between 4 and 6 different program officers before you feel like you've really found a good home for your proposal. So with that, I'm going to end the show so I can see your guys' faces, and I'm happy to answer questions. I can also go ahead and give my email in the chat box, and you're welcome to contact me if you all have any interest in doing so. So thank you very much.
Great. Well, thank you very much, Joanna. That was very informative. I learned a lot about the NSF. I realized I never really understood the missions and the priorities of the NSF. So yeah, that's incredibly useful. What if I could ask you maybe a specific question just to get your take on suitability for NSH funding? So a lot of my laboratory is evolving strongly in the direction of mycology, of really fungal biology, and a lot of it's in the context of immunology and disease. I realize a lot of what I'm doing currently would not be appropriate for NSF funding, but increasingly we're becoming just interested in basic fungal biology, and so with an idea that maybe if we learn more, maybe that might in the future have relevance to pathophysiology to human disease. But anyway, so we're just applications just looking at fungal biology, would that be responsive?
Absolutely.
Potentially.
And depending upon the question you're asking, it could either go to MCB or iOS. A lot of times what people will do that I think is really smart is that they will look at or they will compare different fungi that have different phenotypes. And one or two of those fungi may be clinically important or important to the agricultural field, and then a couple of them may not be, right? So that's kind of a way that what you're doing is using comparative biology to identify mechanisms that are important for persistence or pathogenesis or whatever you call it. And of course, you learn about a pathogen that causes human disease along the way. But once again, it goes back to that broader question of what's important in fungal biology, what makes a fungus robust or resilient or makes it a good pathogen or mutualist. Okay?
Super, thank you. So, and I presume that the thinking is similar for other groups of pathogens, viral, bacterial, et cetera?
Yeah, so for example, you know, we funded COVID-19 research, but a lot of the research that you'll see that iOS funded was looking at mouse and rat coronaviruses, for example, because there are some wonderful rat coronaviruses that infect different strains of rats, and they can either cause a very virulent or avirulent infection. So now you have the ability to look at and identify both host and viral factors that are controlling disease and permissivity, and in a very natural host setting, right? So that gives you a flavor of the types of proposals that do well.
Great. So a lot of some of the immunologists are very much involved in computer modeling, computer sciences, and it's pretty clear that that's also potentially responsive.
Yeah, and in addition, I will say that you should, for people who are involved in modeling, there is a section in engineering that's called Math Bio, and I don't know, you'll have to go look for it, but they're really interested in computational biology, and they're interested in that theoretical side. So for them, they really don't want wet bench research. They want computational stuff first and foremost and at the forefront, with then some experimental biology to test those models that they're setting up.
Right.
And we've funded that before. And once again, you'd want to reach out to the Math Bio program directors to understand what they like in terms of the amount of computational biology versus wet bench experiments.
Okay. And a lot of our additional members of our consortium are really focused on cancer and cancer immunobiology. And so where do you think responsiveness could come in that area? So for example, just fundamental cancer biology?
So it would depend on the question. You could imagine that it could be an MCB if it's talking about dysregulation of let's say genetic of the genome, of expression of the genome. You know, why does a cancer cell have some of its genes dysregulated versus normal cells that don't?
Right.
You could imagine that might be something of interest to iOS if the question is, why are some cancer cells detected by the immune response and resolved, whereas others aren't? Don't know. I mean, if we knew that, oh my God, we could cure cancer, but we don't.
Right.
And yes, people have studied it in humans and mice, but maybe somebody has a great model system in zebrafish or corals that could be beneficial as well.
Right. Very good. Thank you. So a couple of additional questions. So Xiaoyang is asking, can you weigh the relative balance between science merit versus broad impact?
That's a tough one because there's no magic number I can say. I think the best thing to do is to find at least 3 different PIs whose proposals have been funded by NSF and take a look. What I will say is that it is more than just, hey, we're training postdocs and graduate students and undergrads. That's not going to cut it. It really needs to be that you're working to broaden diversity. You're targeting undergrads or high schoolers who are underrepresented, or you are doing science cafes. So it's got to be something more than what you do normally as a PI. But don't worry, we don't expect you to go out and, make a whole new board game that then becomes this huge company. That's not the point either.
Good.
And you can call me and we can talk about that further if you want.
Okay. A couple more just fundamental questions about the application process for NSF. How much preliminary data is generally expected, and at the same time, what about budgets? What is a typical budget for a R01 equivalent type NSF?
I'm really glad that you brought that up. So first of all, with respect to data, it's the same as it is for NSF. You want to show that you can do the technique, or that you have a difference in systems that's worth studying, right? But in terms of the budgets, here's something that you need to understand. For NIH, if you ask for a budget of $100, you get $100. And then NIH usually gives your institution $97 because there's a 97% indirect cost rate, right? But for NSF, it's different. With NSF, if you have $100, you don't get $100. You might only get, $50 or $53 because your institution has set up indirect costs, and those are usually around anywhere between 40 to 60%. So the institution will be taking that part of your funding. So our budgets are much smaller than NIH. Now, in terms of R01s, there's 2 things I want to tell you about. NSF has something called a career award, which is a 5-year award for junior PIs. And those are usually anywhere from $750 to $1.2 million, depending upon what you want to do. We don't have modular budgets. Ask what you think you'll need, and reviewers will review your budget. And because they're other PIs, they'll know if you're trying to do something stupid like order gold-plated pipettement, right? The other thing, though, is that most standard NSF grants are 3 to 4 years, but it can be 5 years as well.
Okay, super. Cynthia is asking about the biology of aging. Is that a topic NSF is interested in?
Sure, absolutely. And once again, depending upon if you're interested in looking at a subcellular or cellular level or an organismal level, would depend on whether you're submitting it to iOS or to MCB. We've funded work with the, is it the naked mole rats that don't have cancer, or that live long. So, those types of things, absolutely.
Super. So Ricardo is asking, what about using knockout mice to explore fundamental mechanisms, for example, immune function?
Yeah, so that's definitely acceptable. It's not that we're saying, no, you can't use mice or C. elegans or Drosophila. If those are if you have a good reason for using those, then that's fine. What I'm saying, though, is that if you send in a grant that says 30,000 people die from disease X each year, and we will be using this mouse model of infection to understand why, that's not really very NSF-y, because I don't understand what your basic biology question is. Your question is, how do we save people from this disease? Not what's the biology that's causing disease. I hope that makes sense. Call me.
Good. Super, thank you. And Fong is asking, if we wanted to understand, look at a question regarding comparative biology, is it better to use a single system or maybe multiple systems, including a range of animals, zebrafish to mice, that kind of thing?
Yeah, that depends on the scope of your research and the question that you're asking. So that would be something where, maybe you and I could have a talk to talk about that a little bit more. I think the important thing is that if you're really interested in evolution, that you need to understand that you really can't understand evolution by just looking at 2 points or 2 different systems, right? That if you really want to look at evolution of a process, you need to think deeply about which animals to look at for evolution, for comparison's sake. And you can call me and we can talk about that. Okay. That was my husband. He's going for a walk. We're still stuck here working from home until probably April of 2021.
Okay, I think we've gone through a lot of basics and we got a lot of questions answered. Unless anybody else has any further input, let's see. So yes, we do have another question. So you say that National Science Foundation is not interested in disease, but we need to understand how to tackle this point or how to design our proposal.
Let me back up. I should say you don't want to lead with, we're interested in this disease that human. So for example, you don't want to say herpes simplex virus 1 causes herpes in 1 out of every 6 people. That's not what we're interested in. What we're interested in is, what are the mechanisms for why some viruses can remain persistent and some viruses do not? How does a virus that is persistent become triggered again? So it's leading with a biological question instead of the human disease. And there are people who will lead with disease questions at NSF that are successful, but it's not human disease, it's coral bleaching. How do corals bleach? And we need to figure this out. And here's how it might be able to help other types of animals and our understanding of disease processes in general. That may take a while to sink in. You're welcome to call me and we can talk about this further, because it's a nuance that is difficult to grasp, but it's important to grasp.
Right. So so my understanding is, correct me if I'm wrong. So my direct disease focus of an application is not typically responsive in the National Science Foundation.
Right.
That's what the NIH is for, correct?
Right.
But basic fundamental biology that might, particularly immunobiology, that might be relevant to disease, that's potentially fair game.
Yes.
Okay.
Yes. And so all in the way you frame things, and so it pays to think about this and to contact your program officers. Okay.
Super. Another question. So, what is a typical success rate? So, for application, particularly the Career Development Award, I would say.
Right. So the Career Development Awards. So what I can say is across bio, it's somewhere around 25%. So it's pretty big or to NIH, right? With career, I think it might be a little bit lower. I hesitate to say anything while we're being recorded in case I am wrong, because maybe something could happen where they take my little snippet. But I think it's lower than that.
Okay.
If you email me, I'm happy to look up that information and get it back to you.
Super. But that's still significantly better than the NIH for most institutes, it sounds like.
The other thing I should tell you guys is that there's no deadlines for most NSF proposals. You can submit anytime you want, which can be a real barrier for some people, right? If you don't have a deadline. Sure. But the beauty of this is let's say you submit when you're good and ready, and when your project is good and ready, and you get comments back, and the reviewers all say, They don't have any data showing this one thing. Well, in the meantime, while your grant has been in review, you get data to show that one thing. You don't have to wait 4 months or until the next meeting. You can turn your grant proposal in the next week.
So how does the review process work then?
So the review process works similarly. The review process, there are some things that work similarly to NIH in that we have panels or study sections that are a large group of people who are all very bright, and we try as best we can to match the expertise of the panel with the content of the proposals. What NSF does that's a little bit different though is that in addition to those panelists, if we feel like the expertise is not there in the panel, we will then reach out to people that we call ad hocs and ask them to do a review from home that is written and that is not part of the process. And they'll send us their written review and we'll use that. And the panelists will also use that to help inform their recommendations to us.
Okay, so the review panels are sort of always on standby. They could get a grant at almost any time. Is that?
No, so the review panels are a little bit different in that they're not a standing panel like NIH. Each time we have a review panel, the program officer will pick and compose a panel to best reflect what's coming in because it's so diverse. What I will say is that we have a GIPRA goal. We are mandated by the federal government to have answers to 75% of all of our proposals within 6 months of receipt. And so that's what we aim for. Usually that means that there are at least 3 study sections or panel discussions per year. I'm not allowed to tell you when they are. It just depends on the number of grants that are coming in.
Okay, thank you. What is the length of the research plan for a career development award?
Yeah, so that's 5 years. I cannot remember the page limit.
Yeah.
It's a little bit different from other ones in that you also are trying to, because you're also talking about how your teaching and research fit together, okay? And how do you plan on using your research in your teaching, and how do you plan using your teaching in your research? So it's kind of a combination of the two. And once again, I think the best thing you can do is go find people who've gotten these career awards in bio in the past 3 years, reach out to them and ask if you can see.
Okay. So if you say you submit and you don't get it first time around, so are you allowed to respond to reviewer comments and resubmit with a modified application?
Right, so what you'll do, this is different from NIH too, you'll get a different proposal ID number. What I suggest to people is that they spend a half a page to 3/4 of a page saying, hey, we submitted this before, here were the problems that were pointed out, and here's what we fixed. Because that way it just shows that you're listening to the reviewers and you're trying to be responsive. You can, and if that fails, you can resubmit, and if that fails, you can resubmit, and if that fails, you can resubmit. You'll get a new number each time, and you can resubmit until the cows come home. Okay. And you'll know that as your research progresses, your grant proposal is going to change over time too.
So there's really no limit to the number of resubmissions?
Nope.
Yeah, okay. All right, got a new question from Olivia. Do you have any suggestions for grad students applying for the GRFP?
That is out of my area of expertise, so I suggest you go contact the program director involved in GRFP.
What does that stand for, by the way?
Graduate Research Fellowship Program, I bet.
Okay.
I could be wrong.
Got it. Okay. We may have exhausted the crowd. So I've run out of questions. Okay. That was incredibly helpful. Again, thank you so much, Joanna. We really appreciate you taking the time.
I know it is a new environment, but be curious, be like a good little macrophage and start sensing the environment and here to reach out to if you have any other questions, okay?
Sounds great.
All right.
Thank you again.
Okay, bye.
Great. Take care. Bye-bye.